This method can help answer key questions in the neuro-rehabilitation field regarding unresponsive wakefulness syndrome and minimally conscious states following severe traumatic brain injury. The main advantage of this technique is that patients with brain tissue deformation, such as atrophy, swelling, enlargement, and shrinking of ventricular spaces, can be assessed in the chronic stage. Demonstrating the procedure will be Tomoki Uchida, a pharmacist, Kazuaki Yokoyama, a skilled operator, Mizuho Kamezawa, a nurse, Shinji Onodera, and Yoshihiro Ozaki, radiological technologist from my laboratory.
Begin manufacturing reagent kits for the automated production of FDG tailored to the synthesizer in use. Set the syringes for the manufacturing reagent kits onto the corresponding syringe drivers in the automated FDG synthesizer. Be sure to use the automatic program to check the mobility of the pumping system. Check the volume of the oxygen 16 and 18 water and the volume of helium, hydrogen, and nitrogen gases. Also ensure that the tap water is under 25 degrees Celsius for primary cooling and under 22 degrees Celsius for secondary cooling. After 1 hour, ensure that air cannot leak from the reagent kit. Set out vials of acetonitrile, mannose triflate, ethanol, and pH buffer solution. Begin preliminary irradiation of oxygen 16 in the cyclotron. Check that 2 to 3 milliliters